into our biology and bioengineering summer camp. In this summer camp, we'll explore the frontiers of biology and bioengineering in 21st century. Let us meet our guest speaker, Dr. Amit Vaish, who will be joining us at the summer camp. Dr. Amit Vaish is a senior scientist at Amgen. He began his graduate studies in bioengineering with Professor Paul Weiss at Pennsylvania State University and subsequently finished his graduate studies at the University of California, Los Angeles. During his graduate studies, he focused on developing biosensors to measure neurotransmitters. Then, he joined the National Institute of Standards and Technology as a postdoc, where he developed biofunctionalized surfaces to explore membrane protein using neutron scattering techniques. We are really excited to have with us Dr. Amit Vaish. See you at the summer camp. Till
Good morning, everyone, and welcome to the day 2 of the Biology and Bioengineering Summer Camp 2021. We are very pleased to have with us today Dr. Amit Vaish. He's a perfect example that how somebody who is core to the engineering discipline could also contribute so immensely towards the field of biology and biomedical area. And he's going to display you not only his recent innovations and work, but also he's going to give you some insights about why it is so important for all of you to get good exposure of biology. So, Amit, the sessions are all being recorded, given that we have not only pandemic but also cyclone issues are coming up in India right now. So, large number of participants are not able to join us online. So, we are making all the recordings available for them. So, we will now invite Dr. Amit to share his screen, and he will take the session forward. Students can ask the question in the meantime on the chat box. We will try to revert back at the end of the session. Amit, over to you now.
Uh, can everyone see my slides?
Yeah. Just make it full screen.
Okay, nice. Okay. Thanks, Professor Srivastava, for organizing this event and inviting me for this wonderful forum. And thank you all for attending this forum. I think this is a very important symposium, partly because we are at the cusp of a revolution, biotechnology revolution, which can in fact solve a lot of the problem humanity is going to face in next few decades and over a century. And it brings the scientific solution to the problems which are imposed on us. And with that, I will start the role of engineering in biotechnologies and what are the challenges it poses and what are the opportunities we have as a scientist, as an engineer. So I would say, and a lot of my colleagues will agree, that this century is the golden age of biotechnology, partly because uh, there are confluence of factors happen, uh, happening as we speak in this century. And these are all because of the development happened in different disciplines of, for example, molecular biology, biochemistry, bioinformatics, and nanotechnology. Now everything is coming together and revolutionizing the field of biotechnology. So I will just take a moment to explain molecular biology, as probably a lot of you know, molecular biology about the genetic code, DNA, RNA. Then biochemistry is about protein, how these proteins are interacting. Bioinformatics field has in fact evolved quite a bit, partly because of the advancement in the computers, supercomputers, and then computational power enhancement. With that, now we got a lot of tools, sequencing tools. So everything is coming together, and the nanotechnology provides the tools to probe all the biological systems are nanoscale. So nanotechnology has developed those tools, whether it's microfluidic devices, it's whether optical system, to probe those things. So now if we combine all these different disciplines, there you see driver for the biotechnology, the confluence of various disciplines, all the things we learn is biotechnology. If we take a step back, probably a lot of you are uh, born in this century and not familiar with what happened in the last century, and you are taking advantage of the all the great thing happened in last century. For example, information technology, internet, fast computers, uh, smartphones. But it all happened, it was all possible because of the physics and engineering blend together, and then you start seeing all these revolution. And in that era, we were still learning about the biology. Biology is a very complex system because you start learning different what are the building blocks, how from molecular level to cellular level to tissue level, and then as a cell, as a whole tissue, how does it operate? And then we think about what things we can do to improve the well-being of humanity in terms of making new drugs, new devices. So now uh, these things are individual pieces are there. Now we are putting things together and moving forward with the taking full advantage of biotechnology. But before that, I'd like to still discuss the central dogma of biology and molecular biology, which is basically starts with the uh, genome, which is the code, and that's basically how these four nucleotides are arranged in that's DNA, and then it's transcribed into RNA, which is the intermediate messenger RNA. How many DNAs are going to be into mRNAs? Is there a lot of uh, steps involved? I'm just providing a very simplified version of this dogma. And then finally, once RNA is translated into protein, and these proteins are mainly made of 20 amino acids. And with the combination of these 20 amino acids, you get tons of different proteins. And these proteins are the molecular building block of life. These are the proteins which makes every process, everything what we do possible, whether breathing, whether our heart functioning, where our bones, everything is related to some protein in the cells which is causing this. And if there is some malfunctioning happens, that's how we get into disease state. And that's the drugs what people take is either trying to activate the protein or inhibit the protein so that one feel better. So the whole drug discovery process is pretty much revolve around this dogma to either tune the DNA or work on the protein, either modulate the protein to make things better. And so that's why it's very important to appreciate this genetic code and the process by which DNA is finally ended into protein. And with with that, I would also like to briefly mention two important things happened uh, in last century. One was the structure of the DNA. Watson and Crick resolved the structure of DNA. And then genome sequencing is happened so that we know how many proteins are encoded in the gene. And then there were a lot of other things happened. For example, because of nanotechnology and microfluidics, we started to know how these different DNA interacts, and then how different proteins interact with each other. So this all system related information we got from genome sequencing. And then the other thing happened was in the getting the structure of the protein is because of the revolution in the X-ray crystallography and high resolution cryo-electron microscope enabled us to, to get the three-dimensional high resolution structure of proteins. And these things are very important because when we are talking about developing a drug, we need to know how where drug is going to bind. And these kind of information, when you know the structure, you can design, okay, these are the binding pockets where possibly drug can bind, and then it can modulate the protein. And if it modulates the protein, what things can happen. So we understand this whole process, how, why things can go wrong and how to undo it back. So, and these were the very fundamental tools, and I would say discoveries which leads towards the drug discovery processes, what we are seeing. And with that, I would like to acknowledge recombinant DNA technology, which started the biotechnology field uh, in 1972. So Paul Berg at Stanford was the first one to discover the recombinant DNA technology. And it's seemingly simple concept that a target gene can be inserted into a circular plasmid, into a plasmid. Plasmid is basically a DNA. So this is the first time where a DNA of one target or one species can be inserted into DNA of different species, and then this can be transfected into any kind of cell. And as a result, now cells can produce protein. So with this technology, recombinant proteins can be produced. And this was the breakthrough. And that's how this recombinant DNA technology started, was the foundation of some of the leading biotech companies like Genentech and my own employer, Amgen, and other companies started coming along this technology. But everything started from this recombinant DNA technology. And with that, a lot of therapeutic proteins have been, uh, companies have started producing. Uh, it could be enzymes, hormones, growth factors, interferons, or monoclonal antibodies. That's a subset of proteins which are used as a therapeutic protein. Most commonly, probably most of you know about is insulin, where someone is diabetic, they need insulin to compensate for the lack of insulin. So these artificial hormones, humulin, are injected, and that can supplement the insulin and save someone's life and improve the quality of life. Uh, so, and the other class is monoclonal antibody, which is used for a lot of therapeutic application. And I will talk about one of the application where this thing is used for the treatment of autoimmune disease. So autoimmune disease, for example, rheumatoid arthritis, ulcerative colitis, and these are the indication where body's own immunity starts attacking the body's organ. And this is a very complicated situation where it compromises patient's health, and ultimately it could lead towards the cancer. So here antibodies are very appropriate therapeutic modality, because what they do is they try to block these immune cells. For example, leukocyte. This is a type of immune cell which is found in the body, and this is pretty active in certain kind of indication.
So
So
Amit,
now,
may I
yeah.
interrupt you? So I don't know if you're showing on the slide, but slide is not moving, it seems. Can you just reshare?
Oh, really? None. What's right now is displaying.
Actually, it is displaying the your title slide only.
Oh, really? Oh boy. Huh.
I think it was still very easy to follow what you just explained, the whole journey. But if you could just reshare the slide. Yeah, now it is working
Okay.
well. Yeah. If you want to just recap quickly, you can do that and then move on to the next slide.
Okay. Okay. So my apologies. So here I talked about the 21st century, the central age, the golden age of biotechnology, and because a lot of different disciplines are coming together and revolutionizing the field of biotechnology. And then I talk about the central dogma of molecular biology. which is basically DNA starts with DNA, four-letter word, and then getting to the protein after translation, which is basically combination of 20 amino acids. It's pretty much like a periodic table, and different proteins can be formed with this simple molecular biology, what nature has created. And we have been able to, as a humanity has been able to resolve this thing. And these are the tools which have been used, genome sequencing and protein structure, to understand. And now we are coming to this with certain other technologies like recombinant DNA technology. Uh, can you still see the slides
Yes,
while I'm speaking? Okay.
correct.
So this DNA technology is the very crucial for biotechnology, and that was the more like starting initiating factor for the whole biotechnology industry. And I was talking about therapeutic proteins, and these monoclonal antibodies, the one thing which is now comprises almost 50% of the biotechnology products in the market. And the reason being that these are very specific. They can, for example, for autoimmune disease, they can block the body's own immunity so that it does not interfere with or hamper the organs or causing any distress to the organ. So now these are the signs. And I just want to give an overview of the antibody because this is very critical to understand how these antibodies, which are found in the nature, can be also used for therapeutic purpose. So these are basically produced by our own B cells, which are immune cells, and the body secretes them so that it can take care of unwanted proteins, cells, or toxin. It is a Y-shaped protein, and its molecular weight is 150 kilodalton. The top things are for antigen binding domain. These are basically what you call a target, or things which protein recognizes, antibody recognizes and binds to it. And then effector is FC is called constant domain. And then it can be designed or engineered to have specificity for a diverse set of antigen. For example, as everyone is familiar with COVID right now, and one of the treatment modality for COVID is having therapeutic antibodies against COVID for the treatment. It can reduce duration of the COVID. Or if someone takes the vaccine, the body produces antibodies against the COVID virus. So that's how antibody is produced, and as now at this point we know that cells are the nature's protein factory, and this is schematic talks about, discusses, illustrates how the antibodies are produced in the lab. It's the proof of concept where any molecule for which we want to produce antibody is injected into a mouse, and then and the cell, which are basically T cell or B cell, they are taken out from the mouse. They are fused with a cancer cell because the lymphocytes or the body's own immune cells, their half-life is very short. In order for this to work as a factory, you want it to be longer duration. That's why they are fused with the cancer tumor cells, and this is called hybridoma cell. This is another breakthrough in the biotech industry to develop the hybridoma cells, and then hybridoma cells are able to secrete the antibodies, and it can be collected, separated, and used for the different kind of application. However, so far I talked about the overview and the research. That's how you can make in the lab. But in order for these things to be delivered to patients, it needs to be manufactured in an industrial setup. And that's how biotech companies make the antibodies in large scale. So in lab, it's mostly you make things in milligrams, but in reality, when you are talking about delivering it to patients, you need to have in kilograms or tons. For that, you need to have a manufacturing process in place. And this is the workflow of the process. So this is divided into two parts: upstream and downstream. Upstream is basically cell culture, where, as I mentioned earlier, that cells are the basically factories for the protein production. So these are the big reactors used to produce the cells. And this the breakthrough came over 20 years is like the yield of the protein production. It used to be 50 mg per liter, like around 2000. Now it's like 10 grams per liter. So you can see here it's probably 200 or 100 times increment in the overall yield happened. And that's where engineering plays a very important role. Engineers play the role in order to optimize the system. Because why do we need optimization? Because it lowers down the cost. These are called biologics, and they are fairly expensive, partly because it's very expensive to make them. They require full sanitization and good manufacturing practice, and all these things require a very clean environment, and all these things are probably one-time use. And that's why one can imagine if you are using with cells, there is a issue of contamination, and all these things make this whole process very expensive. And in order for it to be more viable, this requires further engineering, and this is work in progress where engineers play a very important role. And the second part here is once you get the antibodies from cell culture, you need to purify it with other impurities. That's where chromatography and filtration, all these things play a very important role. In fact, they are 50 to 80% of overall cost. So a lot of engineers, bioengineers, chemical engineers are also playing a vital role in this step. And once these things are purified, they are used for formulation, so that it can fill in the syringe, which is called pre-filled syringe, or packed in the vial. And that's how this goes. That's how the end consumer or patient receives it. So I just walked you through this entire process of biotechnology, and how biotechnology started, and how it looks like in the lab, and how it looks in a manufacturing environment. And then I want to just end up with the final thoughts on the biotechnology, on the future of biotechnology. This is a very nice uh, figure published in Nature by Ray Desai, who is our senior vice president at Amgen. And before he is distinguished professor of biology, chemical biology at California Institute of Technology, where he talked about this different wave of different type of drugs. So the blue one is the first one, which started with serendipity. As a lot of discoveries, a lot of drugs have been discovered by accident. It just so happened that aspirin, which is marketed as Disprin in India, was pain reliever, but it was not meant to be. And there are other drugs like that. It's around 1970, the rational drug discovery approach started, where you know exactly what things we are looking for, and then we started looking for drugs for that particular target. For example, if someone is looking for heart related problem, they started looking for potassium or sodium channel blocker or things like that. And this around 1980, the biotech revolution started when they started looking for therapeutic proteins, such as and humulin is pretty much a mimic of insulin. That was the first drug, first biotech product which came into market. And these are the things. So x-axis is the timeline. Y is the market penetration. It's pretty much how much these drugs are evolving. So you can see that the green one is pretty much like tablet. They reach a plateau. The growth is happening right now here with the green one, and this the biologics. And then the new set of drugs are called multimodality, which are basically the drugs which just don't bind to a target. What they are doing is they are bringing two cells or two proteins together, and as a result of that interaction, magic is happening, and the effects are very pronounced. For example, for cancer cell, one type of modality is bispecific antibodies or bispecific T cell engager. So what this kind of drug is doing is that it is bringing tumor T cells, which are body's own immune cells, and then body immune cells, T cells, are degrading the tumor cell or killing the tumor cell. So that kind of paradigm is shaping the biotech, and I'm very optimistic about it that the future of biotechnology will lie on this kind of technology. I just shared my thought on this thing, but there are a lot of other cool technologies coming along, and with that, I hope that I gave you a snapshot of one aspect of biotechnology, how it is playing a key role in the drug discovery process. And I'd be happy to answer any question if someone has. And with that, thanks for your time, and be safe.
All right, thank you very much, Amit, for this wonderful, overview and journey, how you showed from beginning till where things are currently lying. And before we open up for any questions, I'd like or request you to share with some insight that you had a very lucrative engineering career to begin with, and then you kind of over the time you started learning biology, and then right now you're very much interface of engineering and biology doing work in that area. How
Yeah.
best do you see, like what kind of exposure these children, these students need at the right time, because a lot of things are happening around them right now, but many of them are still quite keen into going for engineering career. So what will be your advice to them?
My advice would be that early on, if you know that interdisciplinary sciences, interdisciplinary engineering, bioengineering, or any kind of thing is much more interdisciplinary in nature. So don't limit yourself with one particular discipline. If even if you are pursuing a career in physics or let's say electrical engineering, that's great, but keep an eye on other things too, how you can apply that field into other avenues. And personally, I am a little biased towards bio-related field because I feel this field has phenomenal potential, because it's still there are a lot of unanswered questions, and there are a lot of technologies which has been developed can be repurposed or adapted for solving the biological problem. So that would be my advice, that at this point, everyone should be uh, uh, open for other ideas, that as a scientist, as an engineer, or could be scientist or engineer, I want to solve the problem. You need to look at the problem. It could be anything, because biotechnology can solve problem from energy to human disease, health, and food security. So you just need to think about it. Uh, these are the tools, and how I can apply to the real world problem, and be passionate about it.
No, that's really great advice. In fact, you echoed very much similar kind of sentiments what Director IT Bombay study stated. Students right now should be very unbiased, explore all the ideas, explore all the fields, just listen to things in with great interest, and explore their interest, and see, like how best they can try to come up with new ideas, right? So let's take a couple of questions. I think Rutuja, you can unmute yourself and ask your question.
Hello, sir. I am
Yeah.
Rutuja. So,
Hi, Rituja.
ask you that multispecificity drugs are now the future of biotechnology because they now can act on many diseases or on many targets in a body. So, can multispecificity drugs focus on cancer based diseases or cancer diseases, which are very important nowadays to cure?
Yeah.
How? I mean, because
Oh,
yeah.
yeah. So that's a great question. Yeah. So in fact, multi-specific drugs, cancers are very well defined because cancer cells are very well defined. Biomarkers are very well defined compared to other diseases. But so far, what we are seeing is that these multi-specific drugs are much more effective in the blood cancer, that kind of indication. Um, Now companies are started to work on the solid tumors, and but we have to see how efficacious it's going to be. The advantage of having the multi-specific is that this is pretty much like you are doing a surgery on it, microsurgery or nanosurgery. That body's own immune system is taking care of the cancerous cells, rather than surgeon going there or chemotherapy where you are killing the cells. Here, exploiting the body's own immune response in killing the tumor cell. Uh, these multi-specific modalities are playing the role of matchmaker, where they are trying to bridge these two things together. Does that answer your question?
Yes, sir. Thank you. Thank you so much for the lecture. It was very nice. Thank you.
Oh, thanks.
Great. All right. So next can be Vaishnavi. If you can unmute yourself, Vaishnavi. Yeah.
Sir, I'm audible.
Yes, you are.
Yeah.
So I have a question that what should be the temperature and pH of analyte in biosensor if it is using an sample in a lab? So what should be the physical condition for that in antibody antigen interaction?
So yeah, that's a great question. I mean, it's pretty much you want to maintain the pH, what's body's physiological, pH is pH 7.5 and and 7.4, 7.5 plus minus, certain decimal point. But mostly when we develop these things, they are tested in phosphate, buffer saline, where it's maintained the physiological salt concentration so that it can mimic as much as possible to the real system. But once because of the time limit, I did not go through the whole process. That once drugs, once you make one drug, it goes through clinical trial. Before that, it goes through animal trial, like mouse study, and then there are 4 or 5 animals it goes through, and depending on the outcome, it goes to human trial. And during that process, you can see if what you said is probably, if these things are not compatible, then toxicity is a big liability. That's why you might have seen that a drug discovery process is a very slow process. Partly because even if you get the drug of interest, molecule of interest, in order for it to go to past reach the human, it needs to go through different animal trial, and toxicity is a big liability. And if something happens, to the animal, it will not go to human trial, and the whole program gets completely trashed, completely shut down. So that's the nature of the biotech industry. That is very high rewarding, but it has a lot of risk, and risk is basically the biology is not very well understood. That's why the future lies in the personalized medicine too. That what Professor Srivastav is doing, omics and bringing all those technology. Once we know more about how individuals are different, then probably we can design better drugs.
Great. The next question could be from Dvija Dave.
Hello, sir. I'm Dvija. Sir, I would like to ask that using recombinant DNA, is it possible like injecting the recombinant DNA into a particular cell and allowing it to grow and forming a new organism? Or maybe can it be possible to revive an extinct species using this technology?
Oh, you want to create a Jurassic Park? Yeah, I mean, Theoretically, yes, but there are other things involved into this thing. I mean, if you think about it, cloning, that's how Dolly, that sheep was created a couple years back. I mean, probably it was created before you were born, 20-30 years back. And yes, one way it can be done, but there are ethical issues involved into it. When you start playing with this kind of thing, changing different kind of species, it causes some ethical concern, and that's a different type of situation. But in theory, yes, it can be done with recombinant DNA technology. It's a very powerful technology. It can be used to clone other things.
Thank you, sir.
We'll just take one or two more quick questions last. So, Zubia Nasir, you can unmute yourself. Yeah.
Yes, sir. Hello, sir. Thank you for the
Hi.
wonderful lecture. I wanted to ask that nowadays people are advocating about avoiding of animal testing. So how avoidable is it? And what is the way around it?
Yeah, so that's
If
a
it is
very
possible.
good question. Animal testing, yes, a lot of ethical issues and other things are involved, but the way around is difficult, but it can be done. So one thing which is emerging field I would say is pretty much a parallel field of biotechnology or offshoot of biotechnology is tissue engineering, like regenerating the tissue. And that is one way to go, where you can regrow the tissue from the cells, and you can have pretty much similar condition, and with microfluidics devices, and you can have vessels and blood vessels, all those things which can mimic the animal model, and that could actually mitigate some of the ethical issues or moral issues arise from the animal testing. When you have a system which mimics with the animal but is not really a living system. But it's a very challenging problem, and the drug industry, pharmaceutical industry kind of deal with it. One of the things we are trying to do in the industry wide is designing the experiment in a very effective way, that it requires minimal animal application, or not at all, other than just rat or mouse, uh, before going for proper clinical trials.
All right, Amit, maybe with one last question, and then if you can just manage a couple of minutes time, you can reply on the chat. There are a lot of questions coming up there, but I think due to shortage of time, we'll probably take just one last question from Tamizini. If you want to unmute yourself and ask a question.
Wonderful talk, sir. So actually for today, pandemic, there is a two-way of healthy and wild type. There will be drug repurposing for another thing, but there is in India, it's a mutational studies. How will that multi-specificity role on mutational studies?
Oh, wow, that's an excellent question. Yes, and this would be actually one of the treatment modality, you can say, which could actually be used for mutation, that you already have known antibodies for, or RNA vaccine for the known target, and then with using multi-modality, you can have like different type of thing for different kind of variants. Yeah, I can see the future here. That's a great idea.
Thank you, sir.
Right,
Yeah,
so
I'm working on that project currently, so I really don't know where to start. So I'm a graduate student, MTech graduate student. So yes, sir. I'm just wondering how well we can do for mutational studies on COVID. So yes, sir.
Mm-hmm.
Thank you so
Yeah.
much for your idea.
very good.
Yeah, that's why I wanted to say that, Professor Srivastav alluded to it, that what advice one would give. And this is the advice, that don't limit yourself with the biotech. This is, sky is the limit. And I can see the best days of biotech are yet to come. And a lot of problems what we are facing can be solved by, at least not all, but majority of the problems can be solved by scientific smart solutions like biotechnology.
one last question from Kanak Sangvi. Long time her hand is raised, so we'll take this
Thank you
last.
so much.
Thank you, sir. So my question is, like, what is the future of biotechnology in forensic sciences? Like, is there any field scope in this field?
Which sciences?
Forensic sciences, like criminals, like
Oh
fingerprints.
yeah, I mean, if you think
Correct.
about it, it is started with DNA fingerprinting, and I mean, in the Hyderabad, that molecular biology lab that played a very important role, and I guess with
CDFD, yeah.
yeah, and Professor Srivastav can give you better insight into it, that with omics and proteomics, it can actually increase the sophistication of that thing, that whole technology. So yeah, I mean, those things are, those tools are there, but I think mainly biotechnology is can be used for health, food security, energy, and a lot of other things which which we can't see now, but after a few years, as things will unfold, we can see the potential.
So Kanak, just to add to here, I think, now the forensic sciences have already been pretty much relying on the looking at, the DNA fingerprint and the molecular biology tools, right? It's routinely being done on all the forensic sites to really ensure that who is the right criminal to do the match. But of course, as Dr. Amit has mentioned, a lot of new potentials are being explored using biotechnology field, right? Where now you can explore a lot of new territories, new drugs, new ways of looking at food security. So there is a huge opportunity for biotechnology to play beyond just a specific, molecular diagnostics. All right, so great, Amit. I think there were a lot of interesting discussions and brainstorming from the students. So thank you so much for sharing your time. I know it is quite late in California right now, and thanks for staying with us and giving this talk. Thank you so
Yeah,
much.
thanks everyone for your time, and thanks for all these great questions, and hopefully be safe everyone, and good luck.
Thank you once again, Amit. So participant will start the
Bye.
next session now.